I've got number 9 posted, but in the meantime, Monday's lecture didn't record.
Mine or yours?
Mine.
Okay.
So could you send me a backup?
This Monday.
I think it was this Monday. Because I went to post it yesterday. Yeah, it was Monday's lecture number 11 on the mitochondrial use of fat. It just didn't record. I'm getting tired of doing the podcast. It's almost better if people do this and then send it to me, then I'm just dead reliable. It's this thing that's the problem. The battery wears out, the other faculty break the wires. All right, so today should be kind of fun. I tell you, I come to class to look at the pictures, and I got some really cool pictures. But much of the stuff that when I was a puppy, we had to just, we theorized existed. With all these new imaging techniques, you can actually show them. So this is, for me, it's fun. So what we're talking about here, and the topic, just to remember the topic we're on, we've learned all about individual muscle cells. We've learned all about how they're powered to get their ATP back after they make a contraction. Where we're a little vague yet is how they work together as a team, right? And this gets into if each muscle cell is the horsey. What we haven't talked about is the driver and the yoking system. How are they hooked together and how are they driven? So this should be kind of fun and a little review of your neurobiology. So that's the brain. These the muscles down here. Important to remember, the term muscle memory is just flat out wrong. It's misleading because you start to think that the motor patterns of activation are stored here, and they aren't. The motor patterns are stored up here. So all motion of muscle, skeletal muscle, originates in the brain. That's what starts it. Unless you're having, well, if you're having epilepsy, it also started in the brain pathologically. And there's a few conditions where the muscles become rogue and go out on their own, but they're pathology. So all normal muscle function starts in the brain. The system is really quite simple, and it's equivalent to the anatomic nervous system, where there's an upper neuron and a lower neuron with one synapse to get to the level of the muscle. And so we typically call these in medicine the upper and the lower motor neuron. So the side view, this is an Alexander Tsiaris. He overlaps scans, x-rays, and art, and you get this wonderful superimposed image. But so there's the central sulcus. If someone wants, get it by lock. Does anyone know? Let's say that where the decision that I want to begin moving comes from. What part of our brain is about forecasting future decision making and deciding what to do next? Up here, prefrontal cortex. So here says, must have coffee, find coho. That's there. It's basically, it's the CEO of action is the prefrontal cortex deciding what the next step should be. Then for motor activity, this area up here is often called the premotor cortex. And in there are stored motor patterns, automatic, things you've learned. Where is coho? Must go downstairs. Access stair descending routine. Everything? Yes. What's that called again? Oh, the premotor cortex. Premotor. So prefrontal, premotor, and then just in front of the central sulcus of the brain, right there, this strip, is all there. Are the upper motor neurons. They actually live in the cortex. I have a picture of them, what fun. They are often called pyramidal cells because they form a pyramid, first described by the Spanish histologist Santiago y Ortega, I can't remember his name. They're wonderful. And these were named for a physician named Betz, so they're often called the pyramidal cells of Betz. And so these are the upper motor neurons, and here's their axons leading down So where must these axons go if they actually begin the sequence that leads to movement? They go to spinal cord, right? That's the exit from the skull. So, I don't know if you guys probably haven't had a lot of anatomy, but this is often called the internal capsule because when you dissect it, it's a thick cabling of nerve tissue. And so it looks like this tight, it's like a cable, the internal capsule. This radiation here, these are images of all the axons leading out of the brain. And notice that this center here, this is the motor cortex, and this is your sensory cortex where information comes back from the body. And notice that this is by far the greatest fraction of material going out of the brain. Something like 70% of the brain's total volume is devoted to movement. You can start to see why, like Jay and I were talking about, why naturally generated human movements in training actually enhance brain function. Just a huge amount of the brain is devoted to moving. All right? So those upper motor neurons in the motor cortex in front of the sulcus, they then send their axons way far away. Okay? All right. And, so here is a spinal cord transection. This is the, did I have this right? Yeah, that's right. This is belly side, ventral. This is dorsal side, right? And so the yellow is ascending and descending axons, yellow indicating the color of myelin. The darkness here in this central portion, these are the second motor neuron, or what we call the alpha motor neuron. It's the lower motor neuron that's going to synapse with the self of the brain, and then it's going to finish the journey by leaving the spinal cord and going to the muscle that it was assigned. Everyone's all tracking this? Did they cover this in MPB 101? Okay, good. Was it as pretty? So let's take a close-up of this area here, because if what I told you is just true, we ought to have axons coming down and then synapsing with a second set of nerve bodies here, and then those nerve bodies will go out to become, and there we go. I'm just amazed you can get pictures like this. So these little circles with the white there would be the myelin sheath, and the center darkness would be the actual axons running up and down the spinal cord, which runs in and out of the board in this transection. Then those will shear over, find themselves a lower motor neuron to make kissy face with. You could pretend maybe that was a synapse, or that was a synapse right there attaching to it. And then this is the motor neuron that's going to erupt a second axon that will then flow out of the spinal cord. You can almost predict, see all these? These are axons leaving the spinal cord, and they're going to coalesce into a nerve, a peripheral nerve. Does that make sense? You see where I am. And I want to make sure that this thing is still bouncing. Okay, I'm going to get a podcast today or die trying. So what do you suppose this is? Just look at it a moment. Well, we have little, there's a yellow core here. It's surrounded by white. This is false coloring, but it's pretty true to what. So these are the myelin sheaths. It's hard to see their interruptions, but there's probably a node of Ranvier right there, a node of Ranvier right there. Do you guys remember the nodes of Ranvier? And what those allow is when you get the electrotonic depolarization of the axon, you actually will get arcing, basically. The electricity jumps. Saltatory, we use the word, en español, como se significa saltar? To jump? Excellent. So the saltatory conduction where the electricity arcs, and this greatly enhances the speed. I was looking at Dr. Bodine has wonderful notes. If you're talking about an alpha motor neuron, the typical motor neuron that controls a muscle, 225 miles an hour is nerve conduction speed. Seems adequate, doesn't it? 100 meters per second, I'm only 2 meters. So almost never is nerve conduction a limiting factor unless you actually break the nerve. Oh, so this is a peripheral nerve. Oops. Hang on. No, now I'm going the wrong way. No one knows what's happening. Got to go back to bed. So this is a peripheral nerve leading out to a muscle. Now, I want to point out one thing, because this is going to become progressively more important as we start to talk about recruitment patterns. Look at the size of this axon, this lower motor neuron axon. See how big it is? Look at this one. You see it? Look at that one. Compare it to that one. Oh, compare that one to that one. They're side by side. Make me a guess about how many muscle cells this hooks up to versus how many muscle cells that hooks up to.
Bigger ones get more.
Yeah. So this is a bigger mother. I'm not sure I'm allowed to say that. And so it takes care of more children. It hooks up into more muscle fibers. This is a smaller mother. It will hook into fewer muscle fibers. And you'll see a huge amount of regulation of force production is down to that simple fact that if you want a whole bunch of force, choose a bigger neuron. Is that okay? And here's some of the figures that these just started to come out when I was in medical school in the late 1980s, where you could actually follow a neuron down. What are these little, they seem to be giving hickeys to the muscle fibers themselves. What are these funny little end things? That's the motor end plate, the synapse on the surface of each. So these long stringy things must be muscle cells, right? Notice, bless you, that a single motor neuron, as I just implied, provides information to far more than one muscle cell. Not all the muscles, not all the cells in a muscle, but a whole bunch of them. And that is starting to develop the concept of what a motor unit would be. Does that all make sense? Okay, I think the next couple of slides focus on this motor end plate, which is really interesting. Not quite yet, soon come. As them say down Jamaica, soon come. Let me see. Yes, we'll come to the motor end plate in just a second. Apparently I want to talk about motor neurons just a little bit more. So, let's review our story today. The upper motor neuron lives in the motor cortex that's right in front of the central sulcus. Its axon comes down, goes through the corona radiata, feeds or drains through the internal capsule of bundling, passes down through the brainstem, out to the spinal cord. You know how I'm always, that's an interneuron. Okay, that's allowed. You can have an interneuron. You can have this axon go directly to the cell body of the lower motor neuron, and this is called the anterior horn of the spinal cord. So an anterior horn cell, an alpha motor neuron, a lower motor neuron, those are three synonyms. And then once the synapse is made, off you go, and we're going to talk about the motor end plate in just a moment. A few things about, since we brought it up, the organization of the spinal cord. These lower motor neurons are on the ventral side, the anterior or ventral horn. Dorsal side, this is all sensory information. So sensory information travels on the ventral side in the spinal cord. Feelings come back, movement go out. Let's see, what do we got here? Oh, we already talked about how, I can make words. The axon of the lower motor neuron is myelinated, just like the one in the spinal cord. It is interesting that this allows, so you have electrotonic depolarization here, and then basically an arcing of electricity to the next node. This greatly enhances the speed of, transmission. I was looking that there are things, I mentioned to some of you, the unmyelinated C fiber, which is basically a bare nerve ending that just sits in the muscle, and it usually gives, it's afferent information, feeling, and it usually gives pain, is what you feel. It's unmyelinated, and so it's much, much slower. If this comes down at 200 meters per second, which seems fairly fast, the C cell comes up at about 2 meters a second. So from my toe to my head, about a second. That's sufficient for pain. Remember, if something burning, I'll have a reflex in my spinal cord that'll deal with it much more quickly. But if I need to know I got a hang toenail, it just doesn't rush. Am I making sense so far? All right, and then as shown, we're going to talk about the motor unit in quite a lot of detail before we get to the end of the lecture. That's where the real meat is. Right now I'm trying to focus on pretty pictures. Notice that each individual motor neuron ends on several different muscle fibers, and one thing it does not do, why can no one, this is science textbook stuff. How could they mess this up? There's one motor end plate per fiber, not 5, not 3. Got to go through and redo all the figures. Maybe I'll just retire. So, another interesting thing about these lower motor neurons is for any one muscle, you'll tend to have the lower motor neurons scattered over several spinal segments. So this is sacral 1, lumbar 7, lumbar 6, lumbar 5, lumbar 4, so the spinal cord. And notice how the soleus, the deep postural muscle in your lower leg, starts to have control, lower motor neurons as high as L4 and as far down as sacral 3. So they're scattered all along here. You might ask why. You could talk about it as simply embryological development, because this is where the neurons that migrated to the area stopped. But you could also think about it as a sort of a, protection. So you get a spinal cord transection right here. Do you still have some control of your soleus? Yeah, about that much. These are out of touch and you'll have a weak soleus, but it will still be under your brain's control. So that's one way to think about why are they scattered. It gives you some redundancy against spinal cord injury. Is that okay? Yeah. Does anyone, I'm not sure if you've had anatomy, if I were to say cervical 3, 4, and 5, what's the next part of the poem? Keeps the diaphragm alive. And so a fairly important muscle in your body, the diaphragm, is innervated by three different cervical segments. So if you have a spinal transection very high, at C2, you can't breathe on your own. And I have to say those people don't survive often, and a lot of them that do don't want to. However, if the spinal transection is at cervical 3, 4, let's say it was at below C4, cervical 4, they will breathe on their own quite nicely. They'll be quadriplegic in terms of their motor function. Am I making sense? So, we talk about the motor pool. If all of these, oh, I see, this was medial gastroc was done on this side, and soleus was done on this side. So that's why there's slightly different distributions. But at any rate, all the lower motor neurons collected together are called the motor pool. And so the motor pool for one muscle, this is the description. And you could actually count the total number of neurons available, and each one of those neurons is connecting to multiple cells, muscle fibers. It's all okay? To finish talking about nerves, then we'll talk about the motor end plate. As we just talked about, a big mother neuron, that's the upper motor, that's the inner neuron, upper motor neurons up here. an interneuron, lower motor neuron. If the axon is very large because the cell body is large, it will tend to connect to many more fibers. If the soma is small and the axon is small, it will tend to connect to smaller fibers. Tracking that? Then an interesting property for those of you that remember your electrical, how many have had their electrical physics? Is that 8B? Does anyone remember? 7? B. Okay, good. This is Ohm's law. Voltage equals amperage times resistance. What we're really considered about or interested in, voltage is a matter of how powerful the electric current is. I is the flow. I is the rate of flow. And so, let's see. As the radius gets bigger, radius gets large, the denominator gets small. I'm solving this for amperage equals V divided by R. Sorry, I'm doing math in my head. And so if your radius is a very large number, what happens to this whole thing? I'm doing it wrong then. Sorry. Never mind. It comes out, you get faster conduction in a, apparently voltage is the conduction velocity. Voltage, amperage, resistance. Really, amperage is the velocity. So I'm a little confused about solving this properly. But it comes out that big neurons, big axons, fiber diameter axon, have high conduction velocities. And I kind of solved that badly. Oh, I know what it is. Wait, come back.
Question?
Yes.
If you have a bigger diameter, that means your resistance.
That's exactly it. So great minds think alike. Termin and I have just reasoned to the correct answer, which is the voltage is fairly constant throughout these. What's happening is the larger the diameter, the lower the resistance drops, and therefore you have more forward flow. Does that make better sense? Some of you may be aware in your general zoology or studying of animals or neurobiology principles, some animals don't do myelination. Instead, they just grow a hella big axon. And the classic example is giant squid, which can be like 16 feet long. They just have these huge axons. You can actually see them with your eye because that's their solution to the problem of speedy conduction. Thank you, Jimmy. If both of us think that, it must be right. Is that okay? So, again, it comes out the same. An alpha motor neuron, these are efferents, so powering the peripheral fiber. Skeletal muscle, top speed, 100 meters per second. Notice there's a lot of other stuff that helps integrate that we'll learn about, I think, tomorrow. Not all of it either. Notice, just as an aside for tomorrow's lecture, at the very end of this, we're going to start talking about afferent. Remember, information coming out of the muscle to inform the brain about how well are things going, has the tendon ruptured, you know, basic information. Someone shot an arrow into the bicep, I don't want to use it anymore. And notice there's quite a bit of different information. This is going to do force and positioning. That's going to do force. This does force. That is touch or deep pressure. There's those unmyelinated C fibers going at 1/50th the speed. So there's a lot of information also coming out of the muscle. Let's not think about it just now. Right now our focus is the alpha motor neuron, which has the largest fiber diameter and therefore has the highest conduction velocity. Is that all right? Good. I hope I didn't confuse you. So let's get down to the neuromuscular junction and say a few things about it. So one, not 6. Oh, I see what they're trying to. There is usually one terminal end plate, which may have different podocytes on it, but it's one terminal ending, and this terminal ending is usually in the midpoint of the muscle fiber itself. Very close. Notice that each one of these forms a classical synaptic cleft. There is a an end plate here, post-synaptic end plate. Does anyone remember what these vesicles are full of that are going to be released if what we're talking about is movement? Acetylcholine. Acetylcholine. Whoever said calcium was quite right that once the acetylcholine binds here and starts up an action potential that runs down the cell, that electricity will be converted into calcium inside the cell. But this is our translational step where we take the nerves, the central nervous system speaks in neurotransmitter. So you have to turn the neurotransmission in the axon into electricity on the surface of the muscle. Then the T-tubule and sarcoplasmic reticulum will translate electricity into calcium. Very good. Good review. What else do I have to say about this? I don't think you need to know a lot, that once the acetylcholine binds, it opens acetylcholine-gated proteins. These proteins allow sodium to leak in, and potassium to leak out, and that sounds to me like an action potential. All right? One thing I will say, Dion, I think you disturbed me saying that somewhere in your earlier studies someone had said, and when people get fatigue, has anyone heard that fatigue originates because you fail in synaptic transmission? Has anyone heard that? They heard it in some... Good. That's probably the one thing that does not fatigue. That the muscle starts to drop out in force production long before the surface, the action potentials on the surface of the muscle, or the acetylcholine in the cleft runs out. So if anyone told you that, I'm just sorry, they're wrong. It doesn't. Maybe they were talking about some unusual laboratory prep where they stimulated it above maximal for 3 days. But in humans, this is the one thing we know doesn't cause fatigue. We started in the 1950s with the idea that that's what made fatigue, and it didn't hold up. Was it you who told me that? Sound familiar? Yeah. Okay. So, again, there are a variety of motor end plates sticking to each. Notice how the actual end plate, there's one of them. Now, I agree these little dots here are each kind of little pseudopodia that interdigitate in a synapse, but you get the idea. Here is a scanning electron micrograph where they shattered and pulled and were able to actually catch. This just amazes me. What do you suppose this thing down here that is very, very long and striped is? That's a muscle fiber. These are probably, if this is the surface of the cell, you're seeing the costamere bridges where the Z disc comes out to grab to the endomysium, and that's the motor end plate. Here's another zoom. Did I show you this yet? No. Again, a scanning electron micrograph with false coloration, so it's easier to see the difference. Here is a alpha motor neuron. Notice how it branches, and each one of these goes, because this one goes there. These two are headed off to something way over here. This comes and makes an end plate right there. It's hard to see where that came from. Apparently it was cut off in preparation, but this goes down. Make sense? Bless you. Salud. Salud. So, the end, and this picture shows us the concept of what is a motor unit. So, a motor unit is the smallest functional level of the muscular system because the brain can actually activate one motor neuron. You can do that. It's the smallest possible motion. And that one motor neuron connects to multiple skeletal muscle cells. And so a motor unit is a single motor neuron and all the fibers it innervates. So there's the spinal cord, lower motor neurons, they come out, they slither all around underneath each other, and if you teased one out, you'd find a single cell body connects to multiple muscle cells. And so this whole thing is called a motor unit. And a really critical concept that we'll see in a moment is the innervation ratio, which is how many fibers per axon. And this is a variable. For instance, the classic one in eye muscles, we talked about how eye muscles have to be fast. Also, if you're, like I said, if you're tracking or you're hunting and trying to get a really good focus on a small target, you're moving your eyeball by fractions of a millimeter. So
So if you have a huge amount of force, you just pull your eyeballs all over, you couldn't ever focus. You need tiny little pieces of force for that slithery little eyeball. And so classically, the only place that I'm aware of that this could happen actually have 4 muscles on a single neuron, that's in the eyes. Tiny little motions. The other end of the spectrum would be anti-gravity muscles. If I'm about to turn on my quadriceps, what am I probably going to do? Probably not going to sign my name, right? I'm not going to thread a needle with my toes. I'm probably going to stand up. And so I need huge packets of force. In quadriceps, you may have a single motor neuron hooked up to 2,000 muscle cells. Because if you turn that muscle on, you probably want a lot of force. Does that make sense? Hey, I'm trying to collect diseases as we go along. I think I might have a really cool disease question at some point. Go back to, I forgot to do it, so let me go back to something that's myelinated. Does anyone know there's a specific disease where usually because of some sort of viral trigger, you get a cold, and then some of the viral proteins resemble some of the surface proteins of the Schwann cell that makes myelination. And so in the aftermath of your cold, you get over your cold, and then like 4 weeks later, your limb gets limp or weak. Does anyone know the name of that demyelinating disease that we see in humans? I'd be very amazed if you did, because it's complex in French. Right now we're having problems everywhere, the flaccid myasthenia that's going around in children. This is probably another disease that's operating like this. We don't know if it's killing motor neurons, in which case it's like polio. Polio works by actually killing the motor neurons in the spinal cord, the anterior horn. This disease that I'm trying to develop, Guillain-Barré, must be French. Guillain-Barré is a demyelinating disease. You'd actually, between polio and Guillain-Barré, both of them will leave you completely limp and out of control of your body. Which one would you prefer to have? Guillain-Barré, because the Schwann cells are not neural tissue and they will grow back completely. So Guillain-Barré is very interesting. That it creates a complete paralysis in whatever lower nerves are affected. So the person may be confined to a wheelchair or can actually be quadriplegic, but they always get their function back, usually within a year. Polio, because it killed these cells, is a permanent condition. The lower motor neuron is dead, and it's not coming back. Oh, yeah, again, sort of developing ideas that, Murphy's Law of the body, anything that can go wrong will. I believe some of you are taking the NPB lab, and you guys actually purposely stopped acetylcholine transmission. What did you use? Turbocurarine, which is a curare derivative, which specifically blocks, and you guys will know better than me, is it blocking acetylcholine release, or is it blocking the receptors? That's what I thought too. The original curare, and I've actually seen curare used in the jungle. When I was a boy, we traveled to South America, and we went way back in baboons, and saw the Native Americans hunting with blowguns. And they actually rolled the arrow of the blowgun in natural curare, and they would shoot the monkey, and then the hard part was you had to follow the monkey until it got paralyzed. That was the really hard part, was you had to keep an eye on it, because they're kind of fast. And eventually they fall out of the tree, and you take them home. Sorry, didn't mean to be gruesome. So innervation ratio is how many of these muscle cells are attached to a single, and that's an important deal. You can actually measure this, which is kind of amazing. What you would do is put a needle, you can do this in a human, and I'm sure it's been done in humans. It would be a little unpleasant, because you have to put a needle into the peripheral nerve, a very fine needle, and cannulate one axon, one motor neuron. Then you turn on that stimulator, And so you're activating that one motor unit, right? Now, if you leave that on overnight, patients in the hospital studying some sort of neuromuscular disease, those cells that are active will run out of glycogen, and those cells that aren't will be good on glycogen. So you take a biopsy after 24 hours of stimulation and discover none of these cells have any glycogen in them. They are all part of the same motor unit, same nerve. Notice also the way that a motor unit, the receiver cells are always scattered across the whole muscle. And I was trying to think about this morning about why would you want that. If all the fibers were next to each other within the muscle, you activate that motor unit, the whole muscle would bow in one direction. You get shear forces. By scattering it throughout the muscle, if you just activate this, the whole tendon is pulled on evenly throughout. So it makes for homogeneous. It also, you need this person really hard playing soccer in the thigh, and all of these die, well, the vast majority of this motor unit still functions. There's a lot, you can just tell, we had a rough growing up because there's so many layers of protection against damage and destruction. Phylogenetic growing up, not, I don't know what you personally do. Is this making sense? So this is specific glycogen depletion to actually reveal a motor unit in a skeletal muscle. Hey, good. If we've gotten to it. So most of that was supposed to be pretty pictures, and now we're going to do about 4 slides of sort of technical physiology, but then it's over. Is everyone okay? Good place for questions? Stretching? Okay. So one of the reasons why it's so important to understand about innervation ratio and the fact that it's variable, and the fact that different sized axons of the lower motor neuron innervate different numbers of cells, it turns out to be one of the biggest descriptions of how much force can you get. So we've several times have asked, how much tension can you get? And if you say, how much tension can I get out of a single muscle cell, one cell, in and out of the board, surface myonuclei. What's the answer? How much force can I get out of this? It's proportional to the total number of actinomycin bridges I can make. Since 95% of this area is sarcomeres, you basically can say the cross-sectional area of a single fiber will tell you how strong it is. With me so far? When you start to talk about a single motor unit, the big difference is, and this comes from original studies, where they started to do this sort of cannulation of individual nerves, stimulated them and measured how much force came out of the muscle, and you got really different levels. So for instance, you stimulate a slow twitch. This is where the misconception that slow twitch muscle is weaker than fast twitch muscle came from, is if you stimulate at the level of the nerve ending, the nerve axon. Notice they stimulate here. They don't have, don't you hate it when they gave me units but no markings? Dumbasses. So let's call this 50 and this 100 milliseconds. So that's a 20th of a second, that's a 10th of a second. Notice that strength here, and remember, these are not single fibers. These are motor units. Yes, sir?
Will a motor unit be all one fiber type?
Perfect. Thank you. Yes. Nature has seen to it that each motor neuron only hooks up to cells of the same type. So what you're seeing here is, if we're talking about strength, which of these two is stronger? This one has a higher peak tension. Motor unit. This one developed force a little slower, and apparently, since the main determinant of strength is cross-section area of muscle, you can already deduce that slow twitch motor neurons are apparently hooked up to maybe half as much area, because you're getting half as much force. Are you following me so far? Okay. So last time we talked about fiber types, which are cell types. And then, is it Moses? Yes? iurea. So if you talk about fiber types, this slow motor neuron will hook up to slow oxidative fibers. This fatigue resistant, notice we're now talking about functional characteristics, not metabolism. You're talking about force generation and speed. So this fatigue resistant motor unit must hook up to fast oxidative glycolytic fibers, FOGs. And so this fast fatiguing motor unit, fast fatigable, hooks up to fast glycolytic fibers. So there is a sort of a parallelity between your motor neuron and your fiber types. But notice that when you characterize motor units, the axon itself, you're characterizing qualities like speed to peak tension and fatigability. So let's look at speed first. Speed-wise, here, someone put on units. Good for them. No, and they're right. So this is a slow a slow motor neuron. It comes up to a peak tension in about 50 seconds. A fast unit will tend to come up in about half that time. So if that was 50, this would be 25. Okay? Notice when you look at the property of fatigability, so you've recruited this is a single twitch, then you turn the electrostimulator to high, recruit that entire motor unit, and just leave it there and go to lunch. You come back and discover it has not fallen off in force production. That would be your slow oxidative fibers driven by a slow and non-fatigable nerve. Yeah?
Does this have to do with anything with the size of the nerve?
Yes.
Well, which one is bigger? Confused.
This would be a small motor neuron hooking up to slow. I have to say most of it has to do with the type of fiber it's hooked up to. But they go together. So the slow oxidative fibers, they just don't wear out. It's why the Kenyan marathoners just keep going and going and going like Energizer bunnies for more than an hour. Go to the other extreme. Fast fatigable fibers, they make a lot of force because they're hooked up to a lot of muscle cells, but they check out rather quickly. If you haven't won the round in 2 minutes, probably why combative rounds are usually 3 minutes long. 2-minute fatigue, 6-minute fatigue, using a lot of this, you're done. Is that all making sense? So motor units are characterized by their speed of force acceleration and then how long can they hold the force. So maybe the last slide, we'll do more next time, but no, I've got to do one slide after this to pull it all together. This is Dr. Bodine's research for which she got her doctorate, and what it's showing you is if you get a huge range of force, if you just isolate a single axon and stimulate it, you might get 10 times the force out of one axon versus another. And it leads, Michael, into your question, how can you be getting 10 times the amount of force? Well, it has to do with how many muscle cells are hooked up to that neuron. So if you have a rather small innervation ratio of 50 muscle fibers, you don't get much force. If you have 400 fibers hooked up to that single motor neuron, which will be a bigger motor neuron, you get much more force. And so this is, again, another way that the brain has an option for deciding how much force should each muscle make for the given task. Right? Picking up your pencil is different than picking up your girlfriend, and you don't want to confuse them. In one case, you'll throw your pencil over your shoulder, in the other case, you'll fall on the ground. So, to summarize, this is a good ending place, and we'll start to talk about nervous afferent information coming out of a muscle next time. But let's take our time and go through this. So this is showing you that slow motor units that hook up to slow oxidative fibers tend to have smaller innervation ratios.
right? See, they're exaggerating, but here's 2 muscle fibers per 1 neuron. Go up to a fast fatigable motor unit that makes hella force quickly and then quits. Then it's hooked up to many more, 1, 2, 3, 4, 5, 6, so they're implying 300% more, and that's about right. Another thing to remember is that the slow oxidative fibers tend to be smaller overall fibers. So these 2 fibers together might be the size of one of these fibers. So you have 3 times as many, and each one is 2 times bigger. You're talking about 600% more force coming out of that than out of that. Does that make sense? This is where the idea that slow twitch fiber is weak came from. But no. No. It's only packaged in smaller bundles. The way I would think about weak and strong would be to measure how much force it makes per unit of cross-sectional area, the quality of each piece of muscle. And if you do that, so these are true based on wiring. If what you do, though, is calculate out what is called specific tension, which is how many millinewtons does a square millimeter of muscle make, you discover that all 3 fiber types have exactly the same specific tension or muscle quality. Okay? Nature bundles them into different groups, and we'll talk more about that next time. But in terms of the quality of each piece of muscle, you'll get the same total amount of force, whether what you have is slow twitch fiber or fast twitch fiber. It's also worth noting that when we say slow and fast in terms of fiber type, that's almost a laboratory description. We just saw that 25 milliseconds to peak force for fast fiber, fast neuron, 50 milliseconds. In both cases, you're moving 5 to 10 times faster than the absolutely fastest human external motion that can be made. So to some extent, people think, oh, I don't have fast twitch fiber, I can't be a thrower. No. What a thrower needs is lots of muscle. And the type doesn't matter because you're going to train your brain to do the motor pattern. And even if you've got all of them going on 4 cylinders, 100 milliseconds is faster than the fastest motion your whole body can make. So you're fine on timing, if that makes sense. So quality of muscle is the same, but nature has bundled them into different packets in order for us to recruit the right amount of force for the task, and that's what we'll start to talk about next time. Okay? Last lecture is tomorrow, 8 o'clock.